Going to cover epidemiology, which is the study of diseases, how those diseases spread around, how we prevent those diseases from being contagious and things like that, as well as the disease process, which is how a microbe invades your body, what happens when that microbe invades your body, and how your body is designed to kind of keep that from happening. So let's go ahead and start with the first four things, the four steps that are involved with a pathogen causing you harm, the disease process. So step number one is contact. The microbes have to be able to come in contact with you, some sort of exposed body surface, so things that aren't covered with, say, a mask or your clothes or something like that. So they're going to have to come in contact with some sort of exposed body surface, usually the nose, mouth, ears, respiratory tracts, usually something like that. Then what's going to happen is the bacteria, the microbe, is going to colonize that area that it invades. So it gets inside of your body, and then it has to colonize. It has to be able to set up shop. It has to be able to grow, and it has to be able to divide inside of your body. The next thing is it's going to have to be able to get inside of the sterile parts of your body. It's going to have to enter in from the parts where the bacteria that are found inside of your body, the normal flora and things, are normally supposed to be. It's supposed to be, those organisms are going to be able to leave that area. They're going to be able to cross the lines of defense, the first lines of defense, like tissue and things like that, your skin, and then they're going to be able to enter into tissues where they're not supposed to be, like the sinus tissues or the brain tissues, things like that. They're going to inner ear. They're going to be able to go to tissues where they're not supposed to be. And once they enter into those tissues where they're not supposed to be, that's step number four, an infection. Those bacteria, the microbe or whatever it is, has now navigated itself to a brand new location where it's not supposed to be. It started actively dividing and causing damage to that particular tissue, and that's the infection part. One of two things is going to happen once that infection starts. Technically three. One, the microbes show up, but they don't do anything. And we'll talk about the concept of a carrier state or a latent infection in a little while. Two, the defenses of your body hold that pathogen in check. Your immune system shows up very strong, very powerful, and it's able to completely destroy that bacteria or hold it at bay. And your immune system eventually will win. The other one is the effects of that particular pathogen are going to overwhelm your immune system. You're going to have some sort of damage to tissues or something like that. The disease process is going to occur. Remember, an infection does not necessarily mean that damage is occurring. It just means something's living inside of you where it's not supposed to be, living and dividing where it's not supposed to be. So that pathogen could cause no problems at all, or it can cause disruption to the tissues, the disease process itself. And once that process occurs, you are going to have cell damage, morbidity, and if that continues, that can be permanent. It could repair itself depending on where it's at, or if it gets worse over time, you can have mortality, which is death. So this is the disease process. It's four steps, and then once the infection process, once the bacteria, the microbe has gotten inside of you, disease will occur. So let's talk about the different areas of the body that harbor bacteria that are meant to keep bacteria, pathogens and things from out of the environment, out of you. So internal organs, tissues and fluids inside of you. So your internal organs, your spleen, your heart and things like that, the tissues of your body other than skin, things that are found other than the skin, liver tissue, heart tissue, stomach tissue, things like that. Fluids like blood and lymph fluid and stuff should be microbial free. There should be nothing living inside of those tissues, those fluids and things. So if there is something there, then it is something to be concerned about. And that's how you know that you have an infection or something that's not supposed to be there. You look for the presence of bacteria that are in those tissues where they're not supposed to be. With bacteria and different microbes get in the tissues that they're not supposed to be in is when the process of disease can occur. So once again, we've talked about these two different types of microbes a lot. You have transient microbes, the bacteria that are on your skin for a very short amount of time. These guys tend to be influenced rather highly by hygiene. You can wash them off by washing your hands, and that's what that concept is for. They live on you for a very short amount of time. you can wash them away. You have residents. These are bacteria, microbes that tend to live on you for a very long time. They become established on your body. They live on you. This is things like yeast or staph bacteria and things like that. They live on you. They're supposed to be there. It's normal for them to be there. They're very stable. They're not so easily influenced by what you do on a day-to-day basis and hygiene-wise and things like that. So the different types of infections that you're going to get are going to occur called an endogenous and an exogenous infection. We'll talk about that in a second. But the bacteria on your skin, the good bacteria, the normal flora, can sometimes prevent bad microbes from growing. We've talked about this concept a lot already. They occupy all the space that the bad bacteria or bad microbes would need to live on. So when they land on your skin or on your body, there's no room for those microbes to grow. So the good bacteria on your skin, the commensalistic organisms, normal flora, and things like that, practice or influence something called microbial antagonism. Good bacteria grow. If bad bacteria shows up, it's not able to do anything there. Sometimes you can have something called an endogenous infection. And this occurs when a normal flora, things like staph infections occur. So staph on your skin is supposed to be there. But if you get staph inside of you, inside of the birth canal or inside of the eye and things like that where they're not supposed to be, this could be a good way to get an infection from a normal flora bacteria. So this is something that happens when you cut yourself, a staph infection and things like that. Endogenous infection. It originates from inside, origination genus, inside endogenous. It originates from inside the body. So when normal flora goes to a place or is introduced to a place where it's not supposed to be, where it wasn't previously, especially if that site was sterile, things like tissues and things like that, that particular organism can cause significant problems because there's nothing supposed to be there. It is a sterile site, and that organism can cause problems. One of the ways that we can easily get our normal flora introduced to us is through the process of being birthed. This is how you pick up your microbes in the first place. You are colonized from the get-go. This is where our resident organisms come from. So one of the ways that you get them, you should be sterile inside of your mother's birth canal, inside of the womb. There should be no organisms there whatsoever other than you. And then when you're born, you break the seal to the birth canal and you travel through your mother's birth canal. And inside of her birth canal are going to be hundreds of different species of bacteria. A lot of different species also defecate during the process of birth, which will also introduce different species of bacteria to that baby's body. So the baby's washed in the mother's normal flora. It's going to sometimes be washed in feces, which can introduce different species of bacteria to that baby's body as well. When the mother breastfeeds, he will come in contact with her skin and things like that, get different microbes from her through the process of drinking breast milks and things. We talked about getting antibodies and things, coming in contact with the dad touching him, the healthcare practitioners touching this baby. Anything that goes on in this little infant's life will introduce different microbial species to it. Some of them will be able to set up shop and grow normal flora like they're supposed to, and some of them are going to be bad pathogens and things like that. So hopefully what happens is this baby will be able to have a very healthy microbiome, microflora, normal flora growing on his body within a couple of weeks to help keep him safe from that microbial antagonism concept. So one of the easiest ways for bacteria and things to get inside of your body is through the skin. And we've talked about this concept a lot, how your skin is meant to keep them out. It's dry, has a lot of acids and oils and things on it. But if your skin's broken, they can get inside. Very easy for them to do so through broken skin. Intact skin, very difficult. Broken skin, not so much. Once again, you're going to have those two different populations of bacteria found on the skin as well as the other types of organs, residents and transients. Residents, they're going to be things that live on your skin like Staphylococcus aureus, Staphylococcus epidermidis, and things like that, Corynebacteria. They live on you all the time. We would expect to find them on pretty much every person. It's not uncommon to find them. And when you wash your hands, it's really not going to matter. Those bacteria are supposed to be there. They have evolved to live on the human skin. So when you wash them away or try to wash them away, it's not going to really matter to them very much. They are evolved to deal with that. They can survive hanging on. They know how to hang on. They're evolved to deal with that process. And then you have transient microbes, and these are guys that are going to be coming onto your tissue through things that you do in a normal daily routine. So you might pick them up out in the environment. You pick up open a door handle. The person behind you didn't wash their hands, and they left some nasty bacteria on the environment on that door handle. You get it on your skin, your hands. You continue out through your day, and those bacteria aren't normally going to be on your skin. And those are things called transient microbes. You can easily wash them away by washing your hands or washing your body and wash those bacteria away. They're not meant to be on your skin. They shouldn't be on your skin. But if you leave them there for too long, then they can cause problems. They're able to breach the skin or find them a way inside the body, some other way. Maybe you're more likely to put them in your eyes or put them in your mouth and things like that. Whereas if you just went and washed your hands, you wouldn't have to deal with that problem. So transients versus residents. Residents are things that are supposed to be there. You expect them to be there. You know what you're looking for when you look for them already, that you know they're going to be there. And then transients are guys, species that come in throughout the day, very easily removed by washing your hands, hand sanitizers and things like that. These guys are more likely to cause disease. They're not supposed to be there. They're a little more likely to be different versus your residents, which are a little more stable. They're less likely to do that so much. Your gastrointestinal tract is a really easy way for different bacteria and things to get inside your body as well. Lots of different environments, different oxygen concentrations, different acid concentrations, and things like that. But it's all open to the environment. It's all mucous membrane, which is a really easy way for bacteria to get through the body. It's moist, wet. That's what they like. Different bacteria species like. So there's also a really easy way to get in through the body. You just easily can go in through being eaten, or somebody sticks your fingers in their mouth and things like that. You can easily get inside the body. You can also inhale different bacteria and things like that through the mouth. Swallow them with food, all kinds of different ways they can get inside of you. Flora of the mouth. Once again, inside of your mouth, there are thousands of different unique bacteria that are supposed to be there. And then ones that are not supposed to be there are going to be the ones that cause you problems. You have this microbial antagonism inside of your mouth. Tons of different environments. The backs of your teeth are different than the front of your teeth with the saliva content, the oxygen content, the little cracks inside of your teeth, the spaces in between your teeth, all kinds of stuff. Create different little unique niches, little tiny micro habitats inside of your mouth, which causes thousands of different species to be able to take up in residence inside of your mouth. So your mouth has one of the most diverse counts of bacterial species within it. So once again, inside of your intestines, a really, really, really good way to keep bacteria out, as well as a concept of microbial antagonism. You have thousands of microbes inside of your intestines. In fact, about half of the dry weight of feces is all the dead bacterial species and things like that, dead bacteria bodies that they've died and you've passed them in your feces. So whatever you eat will be eaten by the organisms inside of your intestines, which then contributes to how you smell. So your diet is contributing to them making different types of gases and things like that, which then you pass through your intestines, and that makes you smell as well. So here's a couple of the different bacteria that you can find in the upper respiratory tract and where they're found. Same thing. Down here, different types of where you're going to find different bacteria and things within the gastrointestinal tract and things like that. So it's very important to keep your normal flora healthy. If you don't maintain your skin with lotions and things like that to keep it dry, the bacteria are not going to be very happy. They're not going to be having a good place for them to live. Their house isn't going to be what they like. So it's not a good thing to keep your skin healthy as well. But you can also make sure that the bacteria inside of your body, inside of your intestines, inside of your stomach and things are also happy. So if you take an antibiotic, we've talked about this concept of broad-spectrum antibiotics causing super infections, things like yeast infections. So they can alter the normal flora of your body as well. So sometimes dietary changes can do it too. You take a massive diet shift. You go from being a carnivore to a vegetarian or something like that, your bacteria inside of your body are used to you eating meat. They're going to have to change to get used to you eating a different style of diet. Disease as well can kill off certain bacteria inside of your body or cause an overgrowth and things like that. So it's very important to keep the bacteria inside of your body healthy so they can accommodate these changes and that they're a little more apt to be healthy when it comes to that microbial antagonism concept. So just make sure you eat a healthy diet and things like that. If you do undertake drastic diet changes, you can sometimes take probiotics to help your body get deal with these changes. This is introducing bacteria that are known to be beneficial to the body that make things that the human body likes. You take them in pill form or you can drink them in probiotic shakes and things like that. And the good bacteria will then be introduced onto the stomach, into the intestines and things like that. Well, then they hopefully will start doing what they're supposed to do, being beneficial to the body. So how do we get a disease in the first place? So you got to find a way to get in to the person. We've talked about how they do that. You got to get attached to your family, or to your family, firmly to your host. You have to use your little fimbriae, their little tiny little hard flagella kind of things, capsules, sticky little gummy surface. You can use hooks if you're a virus, or spikes if you're hooks if you're a parasite. And then you have to be able to survive inside of your host as well. You got to be able to avoid being eaten. If you do get eaten, that capsule is going to be able to keep you from dying. You got to be able to avoid the immune system overall. Lots of different ways they can do this. And then if you can do this, then you're going to cause damage to the person or the host organism as well. And then the whole game is just to get out of your host and go start the cycle over and cause somebody else to get sick. And there's a bunch of easy ways to do that. Respiratory tract, skin cells, you can shed through dead skin cells, fecal matter, fecal oral routes, your genital, sexual, things like that. You can pass through urine and then blood, very easy ways to get out of the body. So let's talk about the concept of true pathogens versus opportunistic pathogens and how the body is some of the different things that are involved with bacteria and different microbes causing infections within the body. So we've talked a lot about this concept of a true pathogen and an opportunistic pathogen. So an opportunistic pathogen, once again, is generally a normal flora, something that if you come in contact with it, your body's immune system's pretty much going to take it and just kind of be able to stop an infection or kill it before it even starts. These guys generally don't cause problems whatsoever unless you have a compromised immune system or your skin's broken or something that allows them to find an environment that they like. So these guys generally don't rob banks, but if somebody left the safe open, they'd more than likely walk inside kind of thing. So as long as you're healthy, as long as you maintain your skin and things like that in good, healthy working order, you don't take a lot of antibiotics that cause yeast candida to overgrow and things like that, these guys aren't going to be causing you problems for the most part. But a true pathogen, on the other hand, is going to be able to cause problems regardless of if you're healthy or not. These guys rob banks for a living. They are meant to come in and knock down safes even if they're not open. These guys only rob the bank if the safe's open and they happen to have been in the bank. These guys drive from bank to bank and knock the safes down themselves. So true pathogens are organisms that are capable of causing disease even in healthy adults with healthy functioning immune systems. They are able to avoid the host defenses, invade the tissues, colonize, and things like that, and able to set up shop significantly easier than our opportunistic pathogens. So the virulence or the virulence factors of a disease is how bad that disease is, or how well, I guess I should say, that organism spreads. So virulence is the severity of that disease. A very virulent strain of the flu is something that's going to spread very well. It's going to cause severe symptoms, very high, likely to have a very high death rate. Whereas a strain of the flu with a very low virulence is going to have a hard time transmitting. It may not have a lot of symptoms and things like that. So the different structures on a microbial surface, the proteins, the toxins they produce, and so on and so forth, that cause the disease to either be really bad or kind of light are called virulence factors. How these little things, these surface proteins, these different enzymes that they produce, different toxins, virulence factors contribute to the overall virulence of a microbe. So these are the weapons available to go fight the war. If you have not very good weapons, you're not going to have a very virulent strain. If you have very good weapons, you're going to have a very virulent strain kind of thing. So let's talk about becoming established inside of an organism. Once you get there, that's the very first step to starting an infection, getting inside the organism, your host. So you can have, like I mentioned already, an exogenous agent or an endogenous agent. So endogenous we talked about. These are going to be normal flora. So these guys can cause a urinary tract infection if you don't wipe properly and things like that, or if you have a staph infection on your skin, you cut yourself and they can get inside your skin and things like that. All kinds of stuff. You can get pink eye if you don't wash your hands properly. Bacteria that were already in or on the body that have gotten somewhere else where they're not supposed to be, and they've caused a problem. That bacteria was already in you, hence the endo part, and it originated from inside of you. An exogenous agent, so these tend to be normal flora. These are going to be mostly our obligate pathogens, our true pathogens and things like that. These are going to be bacteria and different microbes that originate from outside the body, something that you eat, something that you get through contact with a different person and things like that. Could be brought in from a mosquito, all kinds of stuff. So these are the two different types of infections that can originate in and outside the body. But once these guys get established inside of you, then you can have the infection start. So let's talk about the different portals of entry. We've talked about these a lot already. Skin, any type of nicks that break the skin, cuts, abrasion, punctures, things like that. Easy way to get in through the skin. Gastrointestinal tract, also super easy way to get inside the body. You eat them, you drink them, you swallow them. That's an easy way to get inside the stomach. Respiratory tract, you inhale or you breathe through your mouth, through your nose, and you inhale them into your lungs. Also very, very, very easy way to get inside the body. Sexual, displaced organisms from the urogenital tract. Sexual, you guys understand how that works. You put them there. And then displaced organisms would be the concept of like a urinary tract infection from improper wiping. And you end up putting fecal bacteria inside of the urethra and things like that, where you end up with a problem. And then transplacental. There are very few organisms that are capable of crossing the placenta during the process of birth. But if you can, that's a very easy way to infect a new host. The blood is exchanged from through the placenta from the mother to the baby, and that's a very easy way for the baby to become infected through that crossing of the blood, mixing of the blood. So the very first thing to understand about setting up an infection, for an infection to progress, you have to have something called an infectious dose. You have to encounter enough bacteria, enough of that microbe in the environment for it to matter at all. I like to explain this one as a castle. I have my castle wall. 5,000 bacteria show up. I have 5,000 archers on my wall. My immune system is awesome. They will be able to stop that infection before it even starts. That's something with a very that bacteria, that infectious dose was not met. They were not able to get inside of my body. My immune system was able to kill them. Now, if I have 5,000 archers and 14 million soldiers show up, I'm doomed. And that's the concept of an infectious dose. You have to have enough of the organisms, enough cells, enough virus, whatever it is, to be able to overwhelm the immune system, to be able to cause an infection. So the infectious dose varies per organism. It can even vary per strain. It tends to be the organisms with very small infectious doses tend to be causing very severe diseases, and that makes perfect sense. If you need one or two organisms to blow up an entire house, that's going to be very dangerous, versus if you need 50,000 organisms to blow up the house, you're not going to be as dangerous. So this is the infectious disease, or infectious dose down here. Measles. Virus, you need one virus particle to be able to come infected with measles. That's it, just one. It's spread through respiratory droplets. You inhale it, very easy to get inside of your body. That's how much it takes for you to get measles. Now down here, cholera, you have to have over 100 million cells. Anything less than 100 million cells, you literally could drink 1 million cells, and it's not going to cause you any problems. Your immune system, your body can wipe this up. They will not be able to cause you any problem whatsoever. But if you drink more than this, or get them inside of your body, then you will have problems. That's so many that they're able to overwhelm your immune system. These guys down here, they tend to be viruses. Once again, viruses live inside of the cells. They're parasites. Tularemia is also a parasite. Smallpox is a virus. Brucellosis, very small bacteria. So these guys hide. They're very good at hiding, so they're very easy to have, or it's easy for them to have a very low number, a very low infectious dose. Whereas guys down in here, they live in the water. There's really no need to spread. You need to eat them. You're probably going to eat 10,000 cells on one grape, 10,000 bacterial cells of different species on a half of a grape. So it's very easy to eat 10,000 typhoid cells. Kind of hard to inhale measles, 10,000 measles viruses. Chances of you being able to inhale that all at once is pretty slim. So these guys spread by water, very easy to ingest 100 million cells in the water, very difficult to ingest 100 million viruses by air kind of thing. So the way that the organisms are spread really comes into how high the infectious dose is, but really it's just that how much it takes to overwhelm the infectious dose, or the immune system. If you don't have what you need, you will not be able to cause an infection. You could have an infectious dose of 5, you show up with 4, no infection. You got to have 5 or more kind of thing. So adhesion, sticking to your host. This is the very first step of getting inside of your host, other than just finding them in the first place. So we've talked about tons of different ways that organisms use to hold on to their hosts, those fimbriae, the little tiny flagella-like things that just stick to the surfaces. Flagella you can wrap around, glycocalyxes, that sticky capsule concept, or slime layer, you can stick to something. Cilia, you can hold on like that if you're a eukaryote. Suckers and hooks and barbs if you're a eukaryote. You got your parasites and things that hold on to the cell. So the most important part of maintaining your infection and causing infection is being able to stay in one spot. If you're constantly being washed around inside of your host, moved around from place to place, there's no way you're going to be able to successfully get food, divide, or things like that. So you want to be able to establish yourself in one spot. It's kind of like buying a house. And that's what the organism does. It adheres to its host, it buys a house, and then it can just start doing what it needs to do from that location. So once you've established your new house, you've adhered to your new host, you have to survive inside of them. And that initial response from the immune system is just going to be to try to eat them. The leukocytes, the white blood cells, are going to show up and try to phagocytize the cells. So lots of different organisms have ways to prevent this from happening. Antiphagocytic factors, things that avoid phagocytosis. So you can either try to have a capsule or things like that, slime layers that make it very difficult for the immune cells to eat them. It can block them from being engulfed in the first place, or if they are engulfed, it can cause them to just pass through the immune cell. So that organism is able to survive inside of the host, even though the immune system is trying to kill it. You can kill white blood cells. So some species of staph and strep can do that. They can just straight out kill them. They produce leukocidins, leuko meaning white, cidins meaning kill, killer of white cells. So they produce toxins that just kill these white blood cells. So the guy shows up to kill you and you kill him. Really easy way to get around that. So these are some of the ways that organisms can survive inside of different hosts. So once you've survived in your host defenses, you're going to try to get out of the host, get out of the place that you're at inside of your host tissues. And there's lots of different ways that different pathogens can do this. So Salmonella is one of them that has the ability to pull itself into the host tissues. These guys, this particular pathogen is going to invade the intestines. You can see the little microvilli of the intestines down here. And it squeeze itself into the intestines. It will pull itself through the intestinal wall down into the deep tissues of the intestines and force itself into the intestines on the inside, or the body on the inside of the intestines, out into the stomach and fun stuff like that, into the bloodstream. So he can leave the inside of the intestines, which is up here, force himself through the intestinal wall into the bloodstream. So that's a fun way that this particular pathogen can move around from place to place inside of an organism, just by forcing itself through the cell membranes of the intestinal wall, and then through the intestinal wall into the bloodstream. So once again, those virulence factors come into play, how well an organism is causing disease. So we'll talk about exoenzymes in just a second, exo versus endoenzymes. So these are toxins. These are what I'm talking about here, these endoenzymes. How is a bacteria capable of producing a toxin and things like that? So there's two different types of toxins that microbes produce, endotoxins and exotoxins. Both of these are important. We've talked about endotoxins a little bit. We've talked about exotoxins a little bit. We talked about endotoxins with our LPS, that lipopolysaccharide that's found in gram-negative cells. This is a type of toxin that's found inside of an organism. It's either going to be produced inside of the organism itself or embedded inside of the cell wall. So it's not going to be released inside of the body unless the bacterial organism itself is dead. The microbial organism dies. The bacteria is dead. Its little cell starts to degrade, and any of the LPSs, the toxins that are inside of it, will then be released into the body. Those LPSs, those lipopolysaccharides, can then elicit an immune response and cause some damage to the immune system and cause your immune system to freak out. And they can eventually kill you, things like toxic shock syndrome and stuff. Endotoxins are only released when the bacteria dies. The bacteria must be present, grow up his body, grow up the toxins inside of his body. The organism dies, and then once he's dead and his cell is damaged, the toxins will be released. So the organism must be present inside of a host for an endotoxin to be there. Exotoxins. Exactly as the name sounds, this is something that an organism is going to produce and then secrete outside of their cell. This makes stuff a little more interesting. Things like botulism toxin work this way. So you can get food poisoning at a buffet. So how this would work, you have this, let's pretend this whole slide set here is the food. Somebody comes in at the buffet, and they introduce a little tiny bit of bacteria over here in this corner of this food. When they didn't wash their hands, they picked up a little bit of Clostridium botulinum from the dirt. They introduced a little bit of it over here on this side of the macaroni tray at the buffet. Now, it sits there all day under the heat lamp. This bacteria over here grows and grows and grows and grows, and it doesn't really grow past about here. But what it does do is it produces tons and tons and tons of botulism toxin. So the botulism toxin is going to be, even though the bacteria is just here, that botulism toxin is going to be produced, and it's going to penetrate all of the macaroni. So you come in later in the day, you take a big old scoop of macaroni over here. There's no living Clostridium bacteria in it, but it is full of the botulism toxin. You eat the botulism toxin, you eat that macaroni, and then you have botulism. You don't have to have the bacteria present for an exotoxin to impact you. That toxin is still capable of doing its deal even though the bacteria is not present. Diphtheria works the same way, Clostridium diphtheriae, and exotoxin as well. So exotoxins, the bacteria does not have to be present. You can come in contact with the toxin without the bacteria long after the bacteria has been gone, and it can still cause some problems as well. Versus an endotoxin, the bacteria has to be present, the organism must be present, and it's released after that organism dies, kind of thing. So here we go. Endotoxins versus exotoxins. Endotoxins, the organism dies, the stuff's released. Exotoxins, the organism's perfectly healthy, and the stuff is released on the outside of their cells. You can also see the different types of tissues and different types of effects that they have on the body, the different types of toxins and things. Okay, so the disease progression, how a disease works. This is the exact same thing as lag, log, stationary, death phase. Lag stage, in incubation period. You come in contact with a brand new infection. It's growing inside of you, doing its thing. There's not enough of that particular organism there to do anything. You might start to develop a little small cough or a little small runny nose, maybe a tiny little fever towards the end of it. Not really anything to write home about. The incubation period. This organism is just getting ready to cause you some problems. The next stage is called the prodromal stage. You are getting worse. Your symptoms are increasing. I don't feel well. This is going to be I just kind of feel yucky. You don't really know what you've got yet. Your symptoms are getting worse, and the organism growing inside of you is starting to multiply. The next one's going to be the period of invasion. This is when these guys are going to able to cross the part of your body into the getting into the tissues and things like that. The organism's going to grow. Your symptoms are going to get worse and worse and worse. And then eventually what's going to happen is they've done everything they can. They're in the microbes are spread out where they need to be, out through your body. There's nowhere else for them to go. You are as sick as you're going to get, and then you're either going to get worse or die, one of the two. Your immune system is going to take over and beat the infection. You're going to get some antibiotics or whatever, or you won't, and you will die. That's called the convalescent period. Convalescence generally means you will live. Convalescence, I assume that would be death period there if you don't. But this is the exact same concept of the lag-log stationary death phase. This is the phase of an infection. Okay, so localized infections are going to occur if you cut yourself or you get a little infection in that one area. Pretty easy to understand. Localized to that one area because you got a wound there or something, and the microbes that are in that one area will stay where they're supposed to be. It's localized to that one area. You can have something called a systemic infection. You cut yourself on the arm. That organism is able to get inside from the tissues of your arm into the bloodstream and then travel around your entire bloodstream. Your entire body is sick. These tend to be things like viruses that you inhale, and then all your tissues get sick, things like the flu. And then you can have a focal infection. This would be things like strep throat, where that organism starts out living in your throat, in your strep throat, Streptococcus pyogenes, and then it can leave the throat and cause problems in the heart and the mitral valve in the case of scarlet fever. So the organism's carried to a different location by the bloodstream. You have a mixed infection that's caused by more than one microbe at the site, polymicrobial. And this is sometimes difficult to treat. So things like a yeast infection or a UTI at the same time. So E. coli would be causing the primary infection. You've got a urinary tract infection with E. coli. The E. coli has caused the normal flora of your reproductive tract and your gastrointestinal tract down here. Sorry, your reproductive tract and your urogenital tract to become off kilter. The E. coli is causing the initial problems. That's the primary infection. Well, since the E. coli has messed up the normal stuff down here, the normal flora, yeast has started to overgrow, which has caused a yeast infection. And that's something called a secondary infection. So one thing is going to be treated, and then you'll also have to treat the secondary infection as well. So you can have more than one bacteria, more than one microbe causing an infection at the same time. So acute versus chronic, we'll talk about this concept in a little bit more detail in a second with the epidemiology portion of this PowerPoint. Okay, so let's go ahead and move on to epidemiology. So I need to change this in my PowerPoint. This is supposed to be puerperal fever. So go ahead and I'll tell you guys the quick history of epidemiology. Pretty much the very beginning of time, way back in ancient Greece, this guy right here, Hippocrates, knew about a disease called puerperal fever. It's essentially a bacteria disease that occurs when a female gives birth, and then quickly after birth, her uterus becomes infected, and she gets fever, severe pain, and she eventually will die. The purpurea is the lining of the uterus. It becomes infected with bacteria from the process of childbirth. People sticking their hands up inside of the the birth canal and things like that, introducing bacteria that aren't supposed to be there from their hands. So in the most people were born at home back in the day. There weren't a lot of doctors that were going from place to place, so it wasn't really a huge deal. It was, people died during childbirth, but when hospitals started to show up in the late 18th, 19th, 17th, 20th century, people started to not be born at home so much, and they would leave their house and be brought to a hospital to give birth. So once that concept started to happen, diseases of puerperal fever started to show up, or instances started to show up very high. So this is where our little story of the discovery of epidemiology starts. Our very first case of epidemiology. This guy right here, Ignaz Semmelweis, 1841. Pasteur hadn't even put to bed the theory of spontaneous generation, and Robert Koch hadn't figured out that bacteria cause diseases yet. So this guy was a very, very, very ahead of his time. So this guy was from Hungary, and he wanted to be a doctor. So he went to medical school in Vienna, Italy. And he went to medical school in Vienna, Italy, and he got a job at the Laying-in Hospital in Vienna, Austria. In Vienna, Austria, the Laying-in Hospital had two main wards that were dedicated for childbirth. One ward that was staffed with midwives, women that were only meant to give birth to children, and that's it. They didn't deal with any other type of patients. They were not doctors. They only worked with women and their children, and that was it. And the other side of the hospital, the ward was staffed with doctors and medical students. So Ignaz got here, and he walked in his first day, he noticed, oh my gosh, both of these wards, they have the same food, the same supplies, everything's the same, but the doctor's ward of this hospital has significantly higher cases of puerperal fever than the midwife ward. What's up with that? That's kind of strange. So he set forth to figure out what was up, what was going on with this, what was causing this difference. So he figured out that there was a difference, and then he went to go figure out what was up. Why these cases of why so many more women were dying in the medical ward from puerperal fever than in the midwife ward. So he got to thinking about it one day, and he couldn't figure out what the difference between any of this ward was. They had the same food, the same supplies, same everything, same building. The only difference is the doctors. Maybe it's the doctors. Maybe the doctors are doing something that's killing their patients. Maybe it's the doctors. That was his idea. This didn't go over very well. So he kind of started to get a little backlash with this. So he started to push and started to push and started to push. It's the doctors. It has to be the doctors. Something's different. He got a lucky break one day when one of his friends, who was also a doctor, was doing an autopsy on a woman who had died of puerperal fever. His friend nicked his finger with a scalpel and ended up contracting, he was a male by the way, ended up contracting puerperal fever. Now puerperal fever is just caused by a bacteria. It just happens to get inside the body. It doesn't matter where it gets in, it causes the same symptoms. It just happened to be getting in through the birth canal, or during the process of birth for these women. You can get it through other ways as well, like through a cut or something like that, and that's how his friend got it. So he introduced the bacteria from the dead autopsy patient. He cut his finger. The bacteria was introduced, or toxin, whatever it was introduced to his patient's body, his friend's body, and his friend ended up dying of puerperal fever. So Ignaz Semmelweis noticed this, and he figured out that, the difference between the midwives and the doctors is that doctors conduct autopsies. Maybe they are picking up some sort of toxin or something from their hands when they conduct patients, or when they conduct autopsies, and nobody washed their hands at this point in time. That wasn't a thing that people knew to do. And they're carrying that stuff with them to their next patient and causing them to get sick. That's kind of a novel idea. So he put forth this idea of let's just wash our hands and wash our surgery stuff in between patients. Kind of an interesting novel idea. And when he put forth this idea, the cases of puerperal fever in the doctor's ward pretty much went away. The midwives didn't conduct autopsies. They were not getting that bacteria or that toxin on them, and they weren't carrying it from patient to patient. The doctors were, and that was what was going on here. So he put forth this plan of how to stop this disease. People started washing their hand, and lo and behold, it worked. He ended up being exiled from Austria because people didn't really like his ideas of telling people that doctors are dirty. Doctors were gentlemen at the time. They resented that idea. He was forced to return back to his native Hungary. He ended up conducting an autopsy at a hospital in Hungary on a woman who had died of puerperal fever, nicked his finger, and died himself of puerperal fever. So kind of a tragic ending for Ignaz. But Ignaz is the father of epidemiology, and that's kind of why I brought him up. He's the very first person to figure out where a disease was coming from, figure out it was coming from the patient, from the autopsied bodies, how to prevent that disease, wash your hands, and then successfully implement a plan to successfully stop that disease. And that is the study of epidemiology. And this guy right here, Ignaz Semmelweis, was the very first person to do that. So the study of the frequency and distribution of disease is epidemiology. And he's the very first person to do that. Figure out where it was coming from, put forth a plan to stop it, and then actually stop it. So people that study diseases, epidemiology, that's frequency and distribution, are called epidemiologists. Someone that's going to advise local and city and state governments on what to do, If there's a massive outbreak of a disease, what are the patterns of how this thing spreads? How do we control it? How do we prevent it? What do we do? And that's their job. So the modern world has made the study of epidemiology significantly more complicated than it used to be. We have way more people on the globe than there used to be. More people means that it's way easier for a disease to spread around, easier to come in contact with one person to another when there's more of us around. Social unrest. The world has lots of wars at any given time, little governments that are unstable, and tends to be that aid workers don't really want to go into these places. So diseases like Ebola and things tend to just stay in those places, those places with governmental instability or still wars and things like that. Rapid transportation. Here's our big deal right here: airplanes. 200 years ago, you needed to get from the United States to England. You had to go on a boat. If you had a disease that was really bad, it would probably kill you before you even got to the United States and probably infect everybody else on the boat, and they would all die before they got here. Now you can hop on an airplane, go from here to the United States, here to England within about 12, 14 hours, and get off your airplane, and you haven't even developed symptoms yet. You haven't even left the incubation period, and you can take your diseases with you. This is the big problem right here. Global transportation makes disease control very difficult. And then the mass distribution of goods and services. We would never find a banana in Tennessee in 1750. Nowadays, you can go down to the grocery store and buy one for 20 cents. It's easy to move stuff across the entire world very quickly. And sometimes those people, and as well as the goods that they come with, can have stuff on them that we don't intentionally account for. So when those things show up, diseases can come with them. Let's go ahead and talk about a couple of different terms that are associated with epidemiology. These are pretty easy. Endemic, pandemic, and epidemic. So endemic. An endemic disease is a disease that you expect to find at any given time within a population. It's normally there. It's supposed to be there. It's not a surprise that it's there. Things like the common cold, things like the flu. You can go out at any point in the United States, at any point in the year, and take a sample of enough people, and you will find the flu pretty much everywhere you look. You will find it. You will find the cold. It's an endemic disease. It is normal. We know it's supposed to be there. We also know how many cases we expect to find of that disease at any given time. And maybe 10% of the population has a cold at any given time. So we know what that number is just by testing for a really long period of years, long number of years we've tested. We know what these diseases are. We know what they're supposed to be like, just because doctors have reported these numbers for years. So that's endemic, something that's supposed to be there. When you see it, you should not be surprised. You can also have something called an epidemic. And this occurs when it's usually an endemic disease. It can sometimes be a disease that's not endemic to a region, something that's brought in. Mostly it's endemic. Sometimes goes a little crazy on its numbers. So you normally have a little, like 5 or 6 cases of the cold every year in this one town, but all of a sudden you're reporting 70 or 80 cases of the cold in this one town. So that's an epidemic. Way more cases of that particular disease in that area than you would expect to see, and that's an epidemic. An epidemic can be localized, one little town, one little city, one little state, one country, whatever. So it's just more cases of that disease than you expect to see within that measured region, whatever that measured region happens to be. And then the last one you have is a pandemic, where you find the entire world has way more cases of that particular disease than you expect to find. You know that the cold, 5 in 100 people have the cold at any given time across the world. 20 out of 500 people now have it. In the United States, that's an epidemic in the United States. But 20 out of 500 people have it across the world, in each country, and that's a pandemic kind of thing. So these terms are very important. It's also very important to understand what they mean. Very easily misused terms, but technically an epidemic can be one. We have had 0 cases of smallpox in the entire globe since 1977, no wild cases anyway. So an epidemic of smallpox would be 1. Anything better or normal, higher than the normal, which in that case would be 0. You can have sporadic cases of a disease show up from time to time. So here's an endemic disease distribution down here. We know it's supposed to be there. We know what the cases are. You can have an epidemic where you have a small little localized outbreak. More cases in one area. You see one or two in that supposed little area. Now you got a bunch. You can have a pandemic where you have a bunch of little localized outbreaks across the world. And then you can have sporadic distribution where just random cases show up from time to time across the globe. Very difficult to predict random sporadic distribution cases. Okay, so a couple of terms that are associated with epidemiology is the infection rate, or the rate of infection, how fast people are going to become infected. And this is or how many infections that you have. Take the entire population and you would divide it by the number of infections in the area that you have. So if you have 100,000 people in your population and 100 infections, your infection rate would be, there you go, do the math. The less people you have, the higher the rate of infection is going to be, kind of thing. Very easy to figure this math out. That tells you the overall number of percentage, I should say, rate of infection that you could expect to have. So if you know you have this per 100 or 1,000 people, you have 100 per 100,000. Every 200,000, you'd expect to have 200 people, kind of thing. Pretty easy to do this math, also fairly useful. One of the more important forms of these that you guys as healthcare practitioners will see all the time is the morbidity and mortality, weekly report of the morbidity and mortality rates. The morbidity rate is the number of people that will become sick of a disease in that population. So you got 200,000 people in the city. How many of them will become sick? So morbidity does not kill you. Morbidity just means that you are actively sick with that disease. So you can have things with a very high morbidity rate and a very low mortality rate, which I'll get to in just a second. More high morbidity rates just means that it's very infectious, easy to spread from person to person or host to host. Mortality rate, on the other hand, is going to be how many people will die from that particular disease. Fairly easy to understand the difference. Morbidity is how many people will get sick. Mortality is how many people will die. High morbidity rate, you could also have a low mortality rate. Things like the flu. Lots of people get the flu, lots of people get the common cold, but nobody really dies of it. So a very high morbidity rate, easy to transmit, easy to get, but a low mortality rate, nobody dies. And then things like HIV have a very high mortality rate, but a very low morbidity rate. You will pretty much die of HIV. You will pretty much die of Ebola. You will pretty much die of the plague, but it's very hard to transmit those diseases around. They don't transmit very easily, so the morbidity rate of those tends to be very low. So very interesting how these two things play off to one another. Doctors across the country every single week have to report the cases of diseases that they have and they show up in their lab. I had 14 cases of the flu this month, 8 cases of rabies, blah, blah, blah. And this person died of this disease, and you report all of those numbers to the CDC. The CDC reports all that stuff in the MMWR, the Morbidity and Mortality Weekly Report. And this is a combination of all the diseases in the United States that have been seen, how many cases, how many people died, things like that. This is how they know the concept of epidemic, pandemic, and that kind of stuff. What's normally supposed to be there, and then they know when the numbers get higher than we expect to see. Okay. So let's go ahead and talk about the concept of a communicable disease versus a non-communicable disease. So transmission of disease from host to host is a big deal in epidemiology, figuring out how diseases get from host to host and how you can prevent that from happening. So if a disease is termed a communicable disease, that means it's easy to spread around from person to person or host to host through contact. You sneeze on someone, you shake hands with someone, sexual contact, stuff like that. Kissing, whatever, exchange of bodily fluids. Those are communicable diseases. Diseases that are non-communicable, on the other hand, things that you don't have to come in contact with a person to get, would be like malaria or giardia, things that you eat from the environment, or diphtheria, things that come in from the environment from other places. Not diphtheria. Botulism, bacteria and microbes and things that you get from the environment. non-communicable diseases. You're not going to spread those particular organisms from person to person as likely, whereas these things are easily spread from person to person, kind of thing. So we have acute versus chronic. This has to do with the onset of symptoms or the onset of that disease as itself. So something that's termed an acute infection. Acute means it hits hard and it hits fast. Things like the common cold, the flu. Pretty much by about the time that you realize you're sick, you're not really sick anymore. You have 1 or 2 days of feeling terrible. I wish this was over with. And about the time that you start to feel the worst, you're getting better and it's pretty much gone. They hit very hard, they hit very fast, and those symptoms are gone very quickly. And it's called an acute disease presentation. On the other hand, you can have a chronic disease presentation, and these are things that can last your entire life or months at a time. Herpes, HPV, hepatitis, AIDS, things that last for years, that come back over and over and over and over again. Sometimes they will last up until the point of your death. These are called chronic, things that continuously come back over and over and over again. You can also have something called a subclinical or an asymptomatic infection. These are very interesting. The most famous one is Typhoid Mary. She was a cook in New York, Chicago, and places like that. And she had a carrier state infection of salmonella inside of her intestine, salmonella typhi. She would infect people with typhoid fever through her cooking. They would die. She was not impacted by this pathogen. It didn't do anything in her. And they didn't really understand what was going on at the time. They eventually did figure it out, told her to stop cooking. She refused to, still killed some people. They ended up throwing her in jail. But anyway, asymptomatic carriers make an epidemiologist's job significantly harder. These guys actively carry an infection. They are able to shed that infection. The bacteria or microbe inside of them is able to kill anyone else that comes in contact with it for the most part, and cause infection in them. But for whatever reason, this particular person never develops signs or symptoms of that particular microbe. They are able to spread it, but they never show symptoms that they have it. So this makes it very hard as an epidemiologist to figure out who's sick versus who's not. Carrier, once again, is someone who just carries that bacteria inside of them. You can have an asymptomatic carrier, one that carries no symptoms whatsoever. A symptomatic carrier would be someone that's coughing and things like that. You can have a passive carrier, and this is what you don't want to be as a nurse or a doctor. Someone who doesn't wash their hands well, someone who takes contaminated goods, contaminated foods, bedpans, and things like that from patient to patient. This guy over here is doing pretty well with his bacteria disease. This guy over here is a little older. He might be able to his body might not be able to carry or to fight off this infectious disease. So this lady here didn't clean the bedpan well or didn't wash her hands after she was done with it, carried it right over to and then started working on this guy. And she carried that infection from this person to this person, that microbe with her, and this guy might die with that infection. So wash your hands, very important. You guys as the healthcare practitioners to keep in mind. You sometimes can play a role as a carrier as well. So you can have an asymptomatic, as I mentioned. You can have a carrier. Sometimes certain diseases can be spread around during the incubation period. Not every person can do that. Not every disease does that as well. Convalescent periods, you can have convalescent carriers, same thing. Chronic carriers, people who carry this disease forever. Doesn't really cause them too much problems, but they can shed it the whole time. Convalescents, people that can spread it when they're getting better, and they don't get symptoms while they're getting better, and that's the problem. You start to feel better, but you don't have any symptoms, so you're going to go out and do your normal things. You feel better, you seem better, but you're still slightly sick. You can still get people sick as well. So how do diseases spread around, and how do microbiologists and epidemiologists stop this? So this is the disease pathogen triangle up here, the disease triangle. So this one here, the virulent pathogen, is kind of assumed, so I'm going to get rid of that one in word form down here. But these are what we're going to talk about with the disease pathogen, or the disease triangle. These three things have to be found for an organism to be able to cause disease and spread in an environment. The organism has to be able to survive in the habitat that it's found in and able to reproduce there. If this organism is a pathogen that infects penguins in Antarctica, the chances of it being able to impact frogs in the rainforest where it's 95 degrees outside and 100% humidity is very slim, and vice versa, this other way around. Whereas if you have a pathogen that's evolved to impact Gila monsters in the Arizona desert, it'll probably do a pretty good job of impacting lizards in the Sahara Desert. Very similar environment. So the habitat that a pathogen is found in, if it's spread by water or spread by air, things like that, the water has to be warm enough or cool enough or whatever to allow that particular organism to survive in that environment. The organism then has to be able to get from host to host and to be able to survive that process. If I want to get from here to California, I could load myself up in a giant cannon and shoot myself to California. I'm glad I got there, I'm glad I transmitted myself, but the chances of me surviving that are pretty slim. So surviving is the key there. I'm glad you got from place to place, but you have to be able to survive that transmission. And so you can either do transmission from host to host by coughing, making your host do it, shaking hands, sexual transmission, things like that. You can transmit through the water, use a bug or other different things that we'll talk about in a little bit. And then you have to be able to get inside your host. You can either use a bug to do that for you, the bug bites and things like that. You can go through water being ingested and whatnot, ways to get inside your host. So, as an epidemiologist, if you can figure out the way that these three things work in an organism, how it's, what habitat it needs, how it gets from place to place, and how it gets inside, you can stop the spread of that disease by interrupting one of these three things. If you can change the water to be too warm, you can stop that organism from growing. You can, pretty easy on that one. You can kill the bug that it uses to transmit. Boom, done. You can put on a blow-up suit or a mask and keep it from being able to get inside. Boom, done. Pretty easy. If you can figure out how to stop, that's why they call it the triangle. If you knock over one side of the triangle, the whole thing falls apart. And that's the concept here. You have to have these three things are required for an organism to spread, a disease to successfully transmit. And if you can knock out one of these three things, you can stop that disease. So, wherever you are going to naturally find a disease in the environment is called the reservoir. The reservoir can be dirt, soil, it can be a living organism, a bug, or things like that, different mammals and things like that. We are reservoirs of HIV. It lives inside of us, human immunodeficiency virus. Different species can serve as reservoirs. We can too. Sometimes animals can serve as reservoirs. And animals can sometimes spread disease to us, either by directly biting us, things like rabies, or through intermediaries, like a tick. So a tick takes a blood meal from a dog, and that microbe comes from the dog inside the tick, and then the tick brings it to us. So anyway like that, one way or another, that disease originated in an animal, and then it's now in a human. And diseases that originate in animals that transmit to human are called zoonotic diseases. You're more likely to get your dog sick from our diseases than your dog is to get you sick from his diseases. So that's very interesting on that one. We're more likely to get animals sick than animals are to get us sick from their diseases. Now, if you recall the concept of host range and viruses, very big concept plays into this here, which type of viruses can jump the species barrier as well. So there's two different types of transmission when it comes to dealing with diseases: direct transmission or indirect transmission. Direct transmission requires usually host-to-host contact or very, very, very close contact when that host was there. Indirect contact, indirect transmission, does not require a host to have been present or been around within a timely manner. So direct transmission are pathogens, or organisms that exhibit direct transmission, are guys that are not going to be able to survive in the environment for a very long period of time. They're going to die if they come in contact with the high levels of oxygen in the air, or they're going to dry out from the lack of moisture, or things like that. They just cannot survive in oxygen in the environment outside of a host body for very long. So they need to be able to transmit from host to host very easily through the process of just shaking hands, breathing on someone, inhaling respiratory droplets, interacting with infected fluids like blood and semen and things like that. Very, very easy way for this organism to get from one host to the other. These guys, once again, they can't survive very long in the environment. So they have to be able to get from one person to the other, one host to the other very quickly. So that's why they exhibit this direct transmission concept. You can have direct transmission by sneezing, respiratory droplets, and things like that. Some people will argue that this is not technically contact, but if you're close enough with someone to sneeze, for them to sneeze on you, that's direct transmission. Sorry. If you're 10 feet away from someone and they get sneeze on you, you're close enough for them to be directly transmission with you. So they can also sneeze on the table. You can come and rub your hand on the table and pick up that bacteria then. And if it's been such a short enough amount of time that bacteria is still still living on that table and not dried out and died from the oxygen present yet, you're close enough that it's direct transmission. You've been 5 or 10 minutes behind that person. So that's what direct transmission is. It requires very, very, very close contact to another infected individual. So there are two different forms of direct transmission, horizontal and vertical. Very easy to understand the difference. Horizontal is normal, traditional host-to-host. You're sick, you shake hands with somebody at work, and you get them sick. That's very, very, very easy. This is how most people think of diseases spreading, host-to-host. You can also have vertical transmission, and this occurs up here. So horizontal would be host to host. Vertical transmission occurs when the mother gives birth to infected offspring. The offspring become infected from the mother. So that can occur either during the birth process, where the organism lives inside the birth canal. Things like gonorrhea can do this in humans. During the process of birth, the babies pass through the birth canal. The gonorrhea bacteria gets in the eyes and the mouth and things like that, and the baby becomes infected. HIV can cross the placenta. Listeria can cross the placenta. So the baby could become infected before the process of birth as well. So that's vertical transmission. So horizontal is host to host. Vertical is mother to offspring. Then you have indirect transmission. So indirect transmission occurs with something known as a fomite, something in the environment that allows microbes to be able to sustain life. So we've talked about different types of antimicrobial agents. So silver is slightly antimicrobial and things that kill bacteria if they touch them. If something allows bacteria to survive on it, it's called a fomite. Things like doorknobs and stuff, bacteria can live on and things. Inanimate objects that bacteria and microbes can survive on. So you have somebody that doesn't wash their hands very well. They get nasty bacteria all over the doorknob. They could have been there, 2 or 3 hours before you. You come by, you grab the same doorknob, and then you pick up their bacteria from them. That kind of stuff. That's indirect transmission. Lots and lots and lots of stuff can survive on surfaces for a really long time. Doesn't need moisture, doesn't need bodily fluids and things to keep itself wet. So they can survive on the environment for a really long period of time, and you come along and touch the doorknob. It's been hours, weeks, days since another person was there, and you can still become infected that way. So that's indirect transmission. Transmission via food and water. If it transmits by water, filter your water, boil your water. Pretty easy on that one. If it transmits by food, boil the food, store the food properly, things like that. Pretty easy to deal with this one. Our government does a pretty good job of letting us know when outbreaks in the food occur. They test meats, things like ground meats and stuff for O157H7, hemorrhagic E. coli. This is the big deal one that everybody's worried about in beefs. They test every single beef factory, every single thing across the country is tested every single day. Ground meat and things like that, they're all tested for E. coli. Because if they find even one single positive result, the entire batch of meat is supposed to be thrown away. That's how sensitive our government is to this kind of stuff. It's very dangerous. If an organism transmits by air, only the smallest viruses are able to transmit by air. If you can float around in the airstream, you're a very, very, very small little thing. Bacteria are too big to float around in the airstream for an extended period of time. If you cough, they're going to float around in the airstream until they lose their momentum or the air column or whatever the air current runs out. But they won't be able to just float around in the air permanently and just stay suspended in the air column permanently. Whereas viruses are so tiny, they are able to do so. They can float around in the airstream, just the tiny little viruses. You can't control, really difficult, I should say, to control airborne transmission of diseases. You have to filter out all of the air, which is very difficult to do. And you have to breathe, which is also the problem. You can boil the water and things like that. You can use filters for water, but it's very difficult to filter out air. Things that are smaller than air are very, very, very small. So very difficult to deal with airborne transmission diseases. Vector transmission. Sometimes diseases are spread around by insects. And if you spread around by an insect, you are known as a vectored disease. This is vector transmission. So this little flea will take a blood meal from a rat. The bacteria that causes the Black Plague lives inside of the rat. It will go up inside of the flea's mouth in here and clog up his stomach. So essentially what's happening is this flea will never be able to eat. He will permanently be starving. He will be ravenously hungry. And that bacteria has clogged up his stomach, so he won't be able to eat any blood. So he will try to take blood meals from as many things as he can. He will hop around and try to eat, take blood meals, and everything that he goes to take a blood meal from, he will infect with Black Plague bacteria. So that's how that bacteria spreads around from place to place. This is the disease triangle. You have a nice place to live inside of your host here. He carries you from place to place, and he puts you inside of your new host. So vector transmission is the disease triangle. All three things in one. So organisms that transmit via vectors, via bugs and things, they've kind of got it figured out. This is a really easy way to get around the disease triangle. But the problem here is it's also pretty easy to prevent these guys from biting you. Bug spray, squishing them, things like that. You can prevent these guys from getting onto you. So there's a trade-off on that one as well. But nature and wild animals and things, they don't really have hands and or bug spray. So in the natural system, these vector transmissions are very good ways for microbes to get around from host to host and things like that to spread their infection. Okay, so herd immunity, a really big deal on this one. This has to do with vaccination and the concept of disease spreading. If you have a lot of high population that's been vaccinated for a particular disease, if that disease does show up, it won't be able to spread. It won't be able to go from organism to organism. Everybody's vaccinated, so this cow is not vaccinated, you can see. He's sick, he's got a disease. He can't spread it to anybody around him because everybody else is vaccinated. Same over here. Whereas over here, these guys have a lot of unvaccinated cows, so it's very easy for diseases to spread around. You see, this guy had a disease. This guy wasn't vaccinated, but he wasn't sick. But it was very easy for the disease to spread around from this infected cow to that cow. Very easy for diseases to move in low-level vaccination, herd immunity. So herd immunity is the concept of mass vaccination to keep the vast majority safe. Some people cannot be vaccinated. They're allergic to the ingredients in the vaccine, things like that. Some people have different philosophical rejections to vaccines and things like that. Sometimes vaccines just don't work in specific people. We don't really understand why. But those people still need protection. So the vast majority of us get vaccinated. So if a disease does show up, it'll start here. It'll stop before it can even get to an unvaccinated person over here. The disease will never be able to go farther than this one cow. It'll stop with the unvaccinated cow versus reaching over here. The unvaccinated cows that didn't have the disease, it's very easy for them to reach around for the diseases to move within the population. So you have to maintain about a 93% vaccination rate for herd immunity to be a successful thing. Hospital-borne infections are another thing that shows up from time to time, a good way for infections to be spread around. They're called non-sarcomial infections, no sarcomial there. 2 to 10% of patients, so about 1 in 10 to 2, 1 in 10 to 2, 1 in 100 to 10 in 100, 1 in 10 patients will leave the hospital with an infection that they did not come into that hospital with. So this is where you guys come in as the healthcare practitioner with that passive carrier state. You don't want to be the person who carries bacteria from person to person, patient to patient, simply because you didn't wash your hands or carry out good aseptic protocol. So let's go ahead and finish up this lecture with our viral infections. So measles, mirabellobillivirus, very interesting little virus. So once was eliminated from the United States for the most part, it started to make a comeback within the last about 10 years or so due to lack of vaccination, anti-vaccination campaigns. You will hear a lot of people say, oh, it's not lethal, it doesn't kill anybody. Yeah, it doesn't. Not a lot of people die from measles, but it can cause permanent blindness and permanent deafness, as well as a couple of other encephalitis and things like that, swelling in the brain, which cannot be very pleasant. I really don't think it's safe to say that, oh, it doesn't harm anybody. Let's just let it run. No need to vaccinate or worry about measles. Yeah, it does cause problems. Nobody dies, but I don't really want to have my child to be blind or deaf because you guys wouldn't vaccinate your child. So vaccinate your kid for measles. 10 to 12 days after exposure, one single measles virus is all it takes. Very low infectious dose. You look for the Koplik spots, 3 C's, cough, coryza, and conjunctiva. You get little things around your eyes. So that's what they look for. Palliative care, you can't treat the virus. All you do is treat the symptoms, make you feel better. Very, very, very common around the world. Once again, people really don't die from it as often as you can see the case, but people do die from it, and this tends to be associated things with the infection with encephalitis, swelling in the brain, seizures, pneumonia, and things like that. Extra stuff that comes with it. Most of the time, measles is just a very minor fever, runny, coughy nose things. Not that horrible in the symptoms, but when you get into the seizures, pneumonia, encephalitis, it can cause some severe problems. And even if you don't die, you can be left with a permanent, life-altering impacts afterwards. Herpes simplex 1 and 2. Herpes simplex 1 traditionally causes cold sores. Herpes simplex 2 traditionally causes genital warts. Herpes simplex 2, there are thousands of different strains of each of these. Herpes simplex 2, all of the strains, a different disease, herpes. So this is HSV, herpes simplex virus 1, oral herpes. Very small, tends to be a little single, little cold sore on the side of the lip. This is HSV2, herpes simplex virus 2. Genital herpes. You can get it on the mouth. You guys can use your imagination to figure out how that happens. But we've started to see a spread from HSV2 causing oral herpes. You guys can figure that one out. Much more obvious, I should say. You can see more bubbles, more blisters, and things like that. Direct contact, you have to come in contact with the sores for the most part, almost always through sexual contact. 2 to 4 week incubation period, and some people will have these breakouts their entire life. Some people get them once. It really just depends. And vast majority of people on the planet have herpes simplex 1. It's estimated that about 95% of people have herpes simplex 1 within their body, and most people have some version of herpes simplex 2. And then our last one is HPV, human papillomavirus. There's over 200 different species of human papillomavirus. So they cause warts, different forms and different shapes of warts for the most part. Unfortunately, there are a few of them that cause genital warts, and the other unfortunate part is there are a few of them that cause cervical cancer in females. The really unfortunate part is if you have the form that causes genital warts, it doesn't cause cervical cancer. And if you have the form that causes cervical cancer, it doesn't cause genital warts. So you don't know that it's there. That's the problem. It doesn't give you any indication. This guy up here, the tree man of Borneo, he died a couple of years ago, has a very severe form of HPV. He also has an immunocompromisation disease, which combined caused him to grow these massive warts all over his entire body. Most people can shed an HPV infection within a couple of years, and it's estimated that the vast majority of people on the planet also have HPV. It's very easy to get. You can get this through shaking hands with someone. It's very easy to get HPV. I think that's pretty much it, guys. There are really no associated deaths with this one, other than the cancer that comes with the strains that cause cervical cancer. So that's obviously the big deal. Once again, guys, I don't think there's really much else left for this lecture. If you have any questions at all, please feel free to email me. If not, have a good rest of the day.